Okay, everybody, this is Mr. Winchell. This is my first attempt at making a read-through video for the section of notes that I assigned for this week. In the prior part of the chapter, you guys learned about Mendelian genetics, which is simple genetics, where you guys had one trait that was dominant and one trait that was recessive. If the dominant trait was found, or if the dominant allele was found, that trait was always expressed. The only way for the recessive phenotype or trait to be expressed was if both alleles that person carried were the recessive type. However, Mendel's work was very important, and it was in like the 1860s. We have since found many exceptions to his findings, but it is a good start. So the rest of this chapter, which is not very much, and then next chapter, we're going to go into kind of more complicated examples. I want to let you know that I'm going to kind of go through these notes kind of quickly, but you have the luxury of stopping the video, pausing it, rewatching as many times as you need, but fill in the notes as I kind of read through them. So we have since found that some alleles, which again are the letters, are neither dominant nor recessive. We've also found examples of where some traits are controlled by more than two alleles. In all the examples we've done, we've always had one capital letter, one lowercase letter. Sometimes there's more than just two options. And then most traits that you guys tend to think of, like skin color, eye color, hair color, height, weight, they are controlled by more than just one gene. So it's not always just one gene like the pea plant. There was capital T and lowercase t. There was one gene that controlled the height of the plant. For humans, it's much more complex than that. There's many genes that help affect the height a human grows. So I want to talk about two big examples today called incomplete dominance and codominance. Okay, incomplete dominance is where you have two alleles, but one of the two is not 100% dominant over the other one. So in the previous examples in this chapter, we saw again, the big T, little t. If big T was present, the plant would be tall, no matter what. Incomplete dominance is where that big T would not be 100% dominant over the other one. In these types of examples, the heterozygous phenotype, if you guys remember, heterozygous means one of each letter, so like a big T, a little t, the phenotype, which is the way it looks, is expressed, would be a blend of the other two phenotypes. So if this were the case for pea plants, which it's not, and the plant was heterozygous, meaning big T, little t, the pea plant would grow to a medium height. Not tall, not short, but medium. It would be a blend of the other two phenotypes. Okay, so an example of this, which a lot of you guys have probably seen these before, are called 4 o'clock flowers. 4 o'clock flowers come in three phenotypes, colors, red, white, or pink. Now, red and white are the two alleles. You can have the big R, capital R, which it indicates the flower will be red, or a capital W, which indicates it'll be white. Now, I want you to notice we're using two different letters here, and that both letters are capital. The reason both of them are capital is neither one is recessive to the other one. They are both kind of equally dominant, if you will. So the red flower has to have the alleles RR. The white flower has to have the alleles WW. Now, if you take a red and a white 4 o'clock flower and you mate them with each other, if you look at the Punnett square below, you will see all the offspring, which are the 4 blocks, have the genotype RW. This means when they grow, their phenotype, the way they look, will be pink. It will be a blend of the two phenotypes. So not red, not white, but pink, a blend. Okay, so let's say we crossed two pink 4 o'clock flowers. You can see I set up the Punnett square below. Each flower has the genotype RW, so those are on the outside, which again, those red letters represent the gametes, the sperm or egg that those plants produce. And then the box on the inside represent the offspring. So you can see in this case, there would be a 25% chance the flower that was the result of this fertilization would grow red. There's a 25% chance it would grow white, and there's a 50% chance the flower would grow pink. Okay, so now let's take a look at a different example called codominance. Now, the difference between these ones is co means both. Codominance is where both alleles contribute to the phenotype of the organism. So if you look below, you see a white cow, a brown cow, and then at the bottom you see a cow that's kind of white and brown. It has splotches. It shows both colors. Basically, the cell that is producing the pigment for the coloring of the cow's hair, some of the cells produce the white pigment, some produce the brown pigment. Now, I want you to notice it's not the same as the last example. The brown and the white are not blending together to make like a light brown cow. Some of the cells are producing the white pigment, some are producing the brown pigment. So both phenotypes are present on the organism. Both appear simultaneously. Okay, so an example of this is a lot of chickens, their fur color. Their fur color is codominant. They can either be white, they can be black, or they can be speckled, meaning they produce both white and black feathers. Okay, so if the chicken is heterozygous, meaning it has one of each allele, then it will produce the speckled pattern. If it is homozygous, meaning it has two of the same alleles, then it will either be white or black, depending on which alleles it carries, as you can see in the bottom left corner of the screen. Okay, let's look at another example. In cattle and horses, some, not all of them, but some of them can produce a kind of a reddish fur coloring, which would be 2 capital R's. Some can produce a pure white horse or cattle, which would be 2 capital W's. But if you're heterozygous, having 1 R and 1 W, you produce a fur coloring which is called a roan, which you can see that horse in the bottom right corner. And you can see the Punnett square set up below where a red horse and a white horse were mated with each other, and 100% of the offspring would be that roan coloring, kind of some white fur, some red fur. Okay, so let's look at this example. We have a black chicken and a white chicken. They mate together and they produce a speckled chicken. Try to figure out what type of inheritance that is. Okay, compare it to this example where we have a white cat and a black cat and they produce a gray cat. So you can see the one on top, both phenotypes are shown, so that is an example of codominance. The cat example, the white and the black phenotypes blend together, so that's called incomplete dominance. The last couple things I want to talk about in this chapter are multiple alleles. We're really going to come back to this next chapter. I just kind of want to introduce in this chapter. Some genes are controlled by more than just 2 alleles. So instead of just having a big T, little t, in this case we could have 3 or 4 different alleles. So you can only have 2 alleles at one time because remember, we're diploid. We get 1 allele from mom, 1 allele from dad. However, that doesn't mean there's only 2 alleles in the whole population. There could be 5 different alleles. You just have 2 of those 5 possible alleles, or some combination of. So for example, rabbits and chinchillas, their coat coloring is determined by 1 gene, but that gene has at least 4 different alleles. So let's look at the rabbit/chinchilla example right here. You can see on top there's 4 different alleles. Think of how many different combinations you can put those 4 alleles together. Underneath that says genotype. It showed you just 4 of the possible combinations, all of them homozygous. And then below it showed you 4 examples of phenotypes. But there's a whole lot more other genotypes. Like you could put the capital C with the lowercase c, which they didn't show you here. The point being is there's 4 different alleles that control this one trait. An example of this that we will learn next chapter is blood type. Some of you guys may know your blood type. There's different blood groups. The one I'm talking about right here is the ABO blood group. There's 3 different alleles that control this blood group. You can either have the A allele, the B allele, or the O allele. Okay, now remember, you get 2 of those. So you could have 2 A's, 2 B's, 2 O's, or 1 A, 1 O, 1 A, 1 B, or 1 B and 1 O, or so on. There's 3 different alleles that control what type of blood you have. You can see in the bottom, there's that picture right there. I'm not going to go into that in this chapter. I just kind of want to introduce it. But next chapter, we're going to learn all about this and learn how you can potentially figure out your blood type if you don't know it, as long as you can figure out what your parents' blood types are. Okay, last thing are polygenic traits. Poly means many. Most traits that you guys think of, eye color, hair color, skin color, and so on, are controlled by many different genes. So for example, skin color, it's not like you have 1 gene which determines whether you're white, black, brown, whatever. There's many different genes. There's at least 4 that we know of that control skin color, which is why there aren't just 2 skin colors or 3 skin colors. There's a whole bunch of different shades. This is not all-inclusive, but this picture just gives you an example. It shows at least 3 of the genes which contribute to skin color, and you can see, depending on the different combinations of alleles you have for each of these 3 genes, you can see there's a varying shade of skin color. This is just showing you an example, if you know your parents' eye color, what your odds were to have your eye color, which would be on the right-hand side. Okay, I also lastly want to say genes are not the end all be all. I'm sure you guys have heard the saying nature versus nurture. The environment plays a huge role in how your genes are expressed. For example, think back to the pea plants, their height and their coloring and so on. A pea plant's height is not 100% controlled by which gene it has, which alleles it has, like the big T or the little t. It's also highly controlled by the soil and the amount of nutrients in it, the climate, like the weather, how much rain is available to give water to the plant, and so on. So genes are only part of the equation. The environment is the other part. At this point, I'd like you guys to complete the 3 review questions that are found at the end of the notes. And then once you're done with the entire note packet, I want you to scan or take a picture of the note packet, each page, and submit them onto Google Classroom when you're done with them. You have until Monday the 27th by 10 a.m. to turn that in. If you have any questions, let me know.